Welcome to our journey into the fascinating world of molecular biology, where we explore the fundamental building blocks of life. Here's a glimpse of what we'll cover, from the basics of molecular biology to its applications, including DNA structure, genetic mutations, and more. Let's begin with an introduction to molecular biology, setting the stage for our exploration of life's molecular foundations. Molecular biology delves into the processes at the molecular level, crucial for understanding genetics and developing therapies. Discover the pivotal moments and key scientists who have shaped our understanding of molecular biology and genetics. Learn about the central dogma of molecular biology and the techniques that allow us to study and manipulate genetic information. Next, we explore the intricate structure and vital functions of DNA, the blueprint of life. Understand the role of nucleotides in the formation of the double helix, ensuring genetic fidelity. Dive into the mechanisms of DNA replication, a critical process for genetic continuity. Explore how genes encode proteins and how their expression is regulated to adapt to environmental changes. Now, let's delve into RNA and the process of protein synthesis, essential for cellular function. Discover the different types of RNA and their roles in translating genetic information into proteins. Learn about the transcription process and the crucial role of RNA polymerase in synthesizing mRNA. Explore how ribosomes and amino acids work together to translate genetic codes into proteins. We'll now examine genetic mutations and the body's mechanisms for repairing them. Understand the different types of mutations and their potential impact on genetic information. Explore the environmental factors and replication errors that can lead to genetic mutations. Learn about the cellular mechanisms that repair DNA, maintaining genomic stability. Finally, we'll look at the techniques used in molecular biology to study and sequence DNA. Discover the methods of DNA sequencing, from Sanger to next-generation technologies, revolutionizing genomic studies. Polymerase chain reaction, or PCR, is a powerful technique used to amplify specific DNA sequences. It involves repeated cycles of denaturation, annealing, and extension, allowing millions of copies of a target sequence to be generated. PCR is widely used in various fields, including medical diagnostics, forensic analysis, and research, making it indispensable for detecting pathogens, genetic fingerprinting, and studying gene expression. Cloning strategies are essential in molecular biology. Gene cloning involves isolating a specific gene and inserting it into a vector, enabling its replication within a host organism. This technique is vital for studying gene function and producing recombinant proteins. On the other hand, digital cloning techniques utilize computational tools for designing and assembling DNA sequences, allowing precise manipulation of genetic material and facilitating synthetic biology and novel organism engineering. Molecular biology has a wide range of applications that are transforming various fields. Let's explore how these applications are making a significant impact. Medical diagnostics have been revolutionized by genetic testing and biomarker discovery. Genetic testing involves analyzing an individual's DNA to identify genetic disorders or predispositions, providing critical information for disease prevention and personalized treatment plans. Biomarker discovery focuses on identifying biological molecules that indicate the presence or state of a disease, facilitating early diagnosis and monitoring of treatment effectiveness. Biotechnology is at the forefront of innovation with genetic engineering and the production of biopharmaceuticals. Genetic engineering involves the manipulation of an organism's DNA to enhance or modify traits, leading to innovations in agriculture, medicine, and environmental sustainability. The production of biopharmaceuticals involves using living cells and organisms to produce therapeutic proteins and drugs, revolutionizing treatment options for various diseases through targeted therapies. Research and development in molecular biology are driving advances in drug development and agriculture. In drug development, molecular biology techniques are used to discover and test new pharmaceuticals more efficiently, significantly reducing the time from discovery to market. In agriculture, molecular biology emphasizes the use of genetic tools to improve crop yields, resistance to pests, and environmental adaptability, supporting global food security. Thank you for joining us on this journey through the fascinating world of molecular biology. We hope you found the insights into PCR, cloning strategies, medical diagnostics, biotechnology, and research and development both informative and inspiring. See you next time.